interest is genetics, and he's a representative of genetics in APLAR and PSR-SIG. Welcome, Dr. Javed.
Assalamu alaikum, and thank you to the organizers, especially Dr. Babar, for taking the lead in conducting the first ever PSR-SIG meeting, which is something which we all felt should be done. But finally, it's happened, and credit goes to Dr. Babar, Dr. Amjad, and their team at the Fauji Foundation for arranging this meeting. And my task is to talk about genetics in rheumatic disease in Pakistan, but it is primarily on rheumatoid arthritis. The learning objectives are contribution of genetic susceptibility to expression of disease and what role the environmental factors have in pathogenesis. It's a very broad learning objective, and I'll be just touching a very small bit of it. I'll be talking about the research which has been done in Pakistan and published internationally. You can make your judgments about the quality of the research. And what does our data, how does our data compare to the rest of the world? Certain terms, for somebody like myself, DNA was first discovered in February 1953 by Watson and Crick in Cambridge and published 2 months later in a lab which is, I think, about 3/4 of the size of this dais. And that room is still has, in Cambridge, is a historic site, and there's a plaque outside that building. So you need a lot of equipment or a lot of space to do original pioneer work. It's just hard work and sweat and a good idea. So it pays. I'm sure you all understand chromosomes and the genes. Alleles are pairs of genes. You get one from your father, one from your mother. There are certain variations. Most of the time, they have not much significance, but in certain situations, in certain diseases, they become relevant, and that is an area of research. Gene expression is important in expression of the disease. DNA methylation and demethylation. When DNA is methylated, then it is switched off. The genes are switched off, and if it is demethylated, then the genes are switched on. And primarily, it's the role of the mutations, and epigenetics is a study of the environmental factors, and that is something which genes respond over a period of time, how you behave in the environment. And certainly it's relevant in rheumatoid is the role of smoking. That is a epigenetic example of epigenetics, or the environment interacting with genes. Copy number variations, again, are the number of genes which are copied in per chromosome, and there is a data from Pakistan on this topic as well. And SNPs are the bread and butter of research, a single nucleotide polymorphism. The overall heritability of R is thought to be genetically contributed is about 65% in twin studies. The population-based genetic association studies have identified more than 150 RA risk genomic loci or alleles. And these loci collectively account for 15% of the phenotype variations of the rheumatoid arthritis. The primary gene which has been studied so far is the HLA-BRB1 and the shared epitope B1. And that's where most of the research has come and the data is there. But other genes, HLA genes, are the PTNP22 gene, the STAT4 genes, and the others which are in this list. Some, like the STAT4 gene, are important for both lupus and SLE, and so on. And some are more commonly found in populations of European descent, and others are found in the Asian populations. Now, the focus of the research in genetics has been the HLA-DRB1 401 and 404, which is the shared epitope. And 5 amino acids and 3 HLA proteins explain most of the association between the MHC and seropositive rheumatoid arthritis. I'll show you the diagram, what I mean by those 5 amino acids. The variations, the changes in those amino acids lead to expression. Now, this is the study, and I think the slide is the associate the aspartic acid at position 70 versus glutamine or arginine at 70th amino acid position result in a decreased odd ratio of 0.23 and 0.34 for RA patients among English and Spanish population, and an odds ratio of 0.52 among North American populations. DRB1 confers protection. That's Western data. But in the seronegative patients, the DRB1 position, can I have the pointer here? Okay, I need the, okay. So the DRB1 position 11, but in contrast to seropositive RA, which contributes to the 03 and position 9, the carriage of a serine at position 11 increases the risk of developing seronegative rheumatoid, but decreases the risk of developing seropositive rheumatoid arthritis. And glycine at this position 11 is protective for both serotypes. This was one of the ideas when we were doing the students with research in the NUS University is to divide our rheumatoid patients into 4 categories and do their similar kind of work, in which we have a group who are both double positive, a group of single positive each, and then who are seronegative, but it never got to fruition. Now, this is the epitope, and you can see that there's certain positions in here that these are the binding grooves, and the amino acid sequences in different binding grooves lead to different genetic changes and phenotypic presentations. And these are the red balls that you see here. This is where the amino acid sequences, if they change, they lead to increased risk of rheumatoid arthritis or decreased risk, or in the most of it has been studied in the European populations. Now, the non-classical HLA genes have been studied within the HLA region, and the biggest data that I could dig up is from this study by Okada and all in Genetics Art magazine, where they had about over 60,000 cases and controls of Japanese, East Asian, and European populations, and they found an independent association with non-classical HLA gene DOA with seropositive rheumatoid arthritis. And what is the use of doing genetic studies is that you're looking to response to methotrexate and TNF alpha blockers. That's been studied a lot. And I think that could be a focus of research. I know there is one researcher at NUS at the moment. She was doing her, she's from a CMH AFIP, working on methotrexate on diagnosis and screening and also prognosis. Now, the first study that we have from Pakistan is by our very own Dr. Kamran Hamid, way back in 1997. The association of HLA-DRB genes and the shared epitope with rheumatoid arthritis in Pakistan. This was the time when Kamran was training with Terry Gibson, first here at the Aga Khan, and then he went to London Guy's at that time, and the rest of the work was done over there, and it got published in 1997 at the British Journal of Rheumatology. And the conclusion was that in Pakistani population, we have the same shared epitope as it is being reported and studied in other areas. Now, after that, there was a big gap. This was Fazal Jalil. He's done a lot of work. He did his PhD from NUST, and he's not now in Mardan, and he's still contributing for genetics and studying in Pakistan and collaborating with others. And this was published in 2013. There was replication of European rheumatoid arthritis loci in Pakistan, and of 366 patients. My contribution in all this work was myself at the Fauji Foundation at that time, then in my own clinic, then in MH, Brigadier Mushtaq, then in PIMS, Abid was there, and Wajahat and others. They came and they took blood samples. So they just wanted us to give them properly diagnosed rheumatoid arthritis patients. And These were they found out of the genotype the 33 SNPs over there, and 12 of the SNPs were replicated in the Pakistani population. It got published in the journal, well-respected Journal of Rheumatology in 2013. Now, the other study data which now this is again a study coming from the group from the Nurse University. And I tell you that most of the time it is their MPhil and PhD students. They learned lab techniques and they're doing further research. And this was the group, and they looked at the IL-15 gene polymorphism in Pakistani patients with rheumatoid arthritis. I don't know why they chose IL-15. There is literature there. It's an upstream molecule. So it sits at top, and then you have TNF and IL-1 and IL-6, and then at the bottom you have the JAKs. But they thought if maybe any genetic variation at the IL-15 may contribute to susceptibility. But they studied at this SNP1 and SNP2 of the IL-15 gene, and they found no association of that. Now, similarly, another negative study, the TNF-alpha polymorphism of this SNP RS199724, again 134 patients of RA and similarly equal number of matched controls. And then again, they found that there is not much between the controls and the Pakistani populations. Now, this has been studied. The vitamin D receptor gene polymorphism has been studied in other populations. So that group again, they studied at the vitamin D gene polymorphism. Again, this is the SNP, the RS14410 in RA cases and healthy controls, equally matched patients and controls. Mind you, they have taken the blood samples, this lying in their the blood banks, and as the student comes along, the blood samples are taken, and then these experiments are done. Again, negative study, no significant association over here. Now this is again in 2020. This was by Muaz Aslam. He's an interesting. Muaz is somebody who is a graduate of the veterinary sciences from Faisalabad. He did his master's in biological MPhil from NUST and in genetics. And then he's now moved on to Pittsburgh, USA. And he's collaborated with people over there. And every now and then papers get published. And this was the GWAS loci for rheumatoid palpitation in Pakistani patients. And they looked at 58 GWAS SNPs in European, which have been reported in European, and they compared to Pakistani subjects. So there were they divided, there are some related patients and some unrelated patients. That is, you have like a father, brother or sister, or parent and child, and so on. Those were related subjects. And these were the numbers of controls. And of the 58 GWAS SNPs, 14 was reported in Pakistani patients. And so again, the conclusion is something that susceptibility loci are shared between Pakistani and European populations, supporting the role of common genes or common pathways. Now, another paper published about 3 years ago by Muaz and his team, again at Pittsburgh, and 597 rheumatoid cases, 330 healthy controls, and they're looking at the gamma receptor B gene copy number variations in rheumatoid arthritis susceptibility. Remember, I told you copy number variations, how often the gene is replicated in the chromosome. So, and the patients were again from my clinic, MH, and PIMS, and they looked at this FCGR3B copy number variations. And the important finding was that significant association of rheumatoid arthritis risk with less than 2 copies of this was significantly associated, while no significant association was observed in more than 2 copy variation. So if somebody had less than 2 of these copy number variations, their risk increased of rheumatoid, and more than 2 did not. And this copy number variation less than 2 was strongly associated with anti-CCP, with the odds ratio of pretty significant 2.56. while only a trend of association was observed with the rheumatoid factor. So it's more to do with anti-CCP positive patients. Now, this is another the study which got, this is an abstract which was presented at the Genetics SIG 2 years ago in Singapore by Dr. Binishali. She's at ComSets. She has done a lot of work. This is one I'm presenting. That's her experiment that I'm presenting. And these are the 300 or so rheumatoid arthritis cases and healthy controls. And they looked at the genotyping of this SNP, RS1154946. And basically what they showed was there was no significant association to confirm a protection again was done by. There's a protective against the role of fever as a risk modulator in Pakistani population, but there were no significant interactions found between this SNP and gender and smoking to alter the genetic susceptibility. Smoking is an important risk factor, and it has been studying in genetic susceptibility if you know the smoking status of your patients. But in the other experiment that she did and I presented, that's her work. It's just me just presenting her data. The interaction analysis of IL-17 and IL-17 genes in rheumatoid, the impact of genetic variants and smoking disease. Here that we discovered was that there was significant association of increased susceptibility of this SNP, and this SNP. This SNP gives you protection or decreased susceptibility and no association of these. So these like 5 SNPs were studied. 3 gave increased risk with smoking. The other 2, one was protective and the other was not important. Now, this is another genotype that's been studied. And this was published in the same session. Fazal Jalil, who has published a lot, also presented his data on 300 patients and match controls on this SNPs. And there was again this. He did this is the first one. He did microRNA work. This is the first time microRNA has been presented, but this one showed no association. Except that this SNP RS291064 might have a protective role with pathogenesis, but this one, which was of interest, did not differ between cases and controls, so it doesn't lead to increased risk. And finally, last year at in the Japanese meeting in 2025, our group chair, Sulaiman Khan, presented this paper on gene therapy. And again, Fazal Jalil presented this paper on therapeutic targets within IL-2 and IL-3 genes. So that was his paper. I didn't go there, but these 2 papers were presented by Pakistani SIG in Japan last year. So at the end, I would just like to say that the research of genetics in Pakistan is a collaboration between the clinicians and biosciences departments of the institutes. It is plausible, it is doable, because these universities have funding, they have students, and they're looking for well-characterized patients, which fortunately in our specialty are there. We have long-term follow-up, our patients are well documented. So they find it easier to collaborate with rheumatologists than perhaps with other specialties. So we can, if we have, reach out to them, and there are students who are doing MDs and PhDs in physiology, in other biological sciences, in biology, and they have the tools and they have the kits or the measurement. So if you have some interesting idea, then it's just a matter of sitting together with them and maybe having a cup of tea and discuss what the idea is. What you could see is that the way the research is directed is determined by the departments themselves. Whatever there is very little discussion where we should go. Our input is minimal. All we do is we give them, this is our student, this is his or her research thesis, and please help them complete this thesis because they need 300 patients. So that's where we come in. But I would rather that we have a strategy or an aim that what we want them to study. If you ask me, we focused more. Yes, susceptibility. People have looked at susceptibility. Yes, it's interesting. You get your paper published, you get your impact factor journal, आपके CV अच्छा हो जाता है. Your CV is good, but it doesn't add a lot to the practice in real practice. What we would like is, yes, screening again. Yes, it is interesting. We have many families. I have a family of which out of 8, 7 sisters have seropositive rheumatoid. Another family with parents having are rheumatoid and the children have JIA, and one of them is an angst. So we have these clusters of autoimmune diseases within in everyone in each department in each practice, and they are a gold mine if in terms of genetic research to run. But I would say that pharmacogenomics, where response to methotrexate, response to side effects to TNF alpha blockers, should be the focus of a research for us, and we need to categorize patients and then these are the groups of patients. You can take the blood sample, do your experiment or do your research. We have just briefly very little data on SLE. The only thing I could find was one paper on the genetics of SLE in 2011 from Punjab University, nothing else. On SPA, 2 papers that I could find. One was by our very own Brigadier Tahir Aziz in late 90s when he studied HLA-B27, and again in 2022 by the group in AFIP again in MH, Brigadier Amir Fakar and his students. They published another on HLA-B27, and there is work on, there's one paper published in my Google search on psoriasis from Lahore, but not on psoriatic arthritis. So I've just dwelled on what is the idea of priority. There's a genetics has got just 2 members, myself and Suleiman, who's in Ireland these days, but he heads the group, and he's now the APLAR SIG chairman. So whatever we have contributed, that's led to Pakistani Rheumatology Society or Pakistani genetics work being recognized, and now we are leading the APLAR group. So, but I would want more of you. I hope it stimulates or generates interest to join this SIG, and maybe we can build on this what has been done so far. Thank you very much.
Thank you so much, Dr. Javed. I'm sure a lot of us would be interested in genetics from now on. Our next speaker is Professor Dr. Uzma Rashid. Dr. Uzma is among the first generation of rheumatologists trained within Pakistan to the CPSP Fellowship Program. She is currently the head of Department of Rheumatology at TIMS. Her principal area of clinical and academic